In this short video, I am going to discuss the Transfection One Label with Transmitted Light protocol, which is part of the cell biology protocol collection. The protocol is designed to measure the proportion of cells in transmitted light images expressing a fluorescently labeled recombinant protein. First, we're going to select the protocol by going to the count side rivet. Next, we'll press the protocols button, choosing the cell biology collection, and then scroll down to the Transfection protocols. For this one, we'll be using the One Label and Transmitted Light protocol. Next, all protocols include a demo image or image set. In this case, simply go to the Reset button, press the Load Demo button with Open Demo Image selected. I'm going to adjust the background a little bit by selecting the Alexa floor channel, bring that a little bit up. You also have the option, especially for transmitted light, to choose the background image, in this case, the bright field, and adjusting that. This will make no difference in the analysis of the protocol. Next, we can press the Sync button to load the image channel names. In this case, the cells will be bright field, and the labels, or the Transfection label, will be Alexa Floor 633. And now we can test the settings for the protocol with the demo image, or if you're using your images, you can set it accordingly. First, make sure the interactive is selected and checked, and press the Next Step button. For this step, we'll be segmenting the transmitted light cells. I'm going to turn off Show Preview Mask for a second so you can see how well they're selected. And I'm going to zoom in a little bit. There are a series of controls available. The first is adjusting the threshold. Going lower, you'll select more. Higher, you'll select less. Then you can dial this in, or you can type in a number. There's a smoothing filter, which can be used to run a low pass filter on there. Bring that back a little bit. Cluster size is setting the size of the cluster in order to determine what the background correction factor should be here. If there's any illumination, we'll correct for that. And then finally, a close filter. What the close filter is doing is filling in these gaps. If I go lower, it's a smaller kernel size, and larger, it will fill in large blocks. Again, this allows you to dial it in based upon your sample and how well we can segment the transmitted light cells. Let's bring it up a little bit. Here we have Show Preview, and you can turn that off and see how well you're doing. When you're done segmenting, press the OK button. Now we can look at our results. There are segmentation options where we can set a minimum area size, using histogram selection to filter out areas you don't want to measure. I'll press the OK. Next, we have clean borders, where we can filter out any cell touching the edge of the image. Fill holes. Right now I'm not going to do that because I don't want to fill these gaps where there is no cell. And reference size. Reference size allows us to do an estimated cell count. Just because of the difficulty of segmenting these cells in bright field, we can use it to give an estimated size. Here, let me take this one. And so the idea is we can select a cluster, prompting you to select this particular cluster. In this case, choose that one. There are clearly two defined cells in there. Press OK, and then you tell it how many cells are there. And what that will do is give an estimated size of the cell to generate an estimated cell count, just because sometimes it's difficult picking up the number of cells in transmitted light. Next, since I was making segmentation option adjustments, I'll press the OK button, and that will update the analysis of the image. We split everything according to how it's made my adjustments. Press the Next Step button. And now we'll segment the transfection or the marker channel. Here we can see it. They're pretty well defined. We can always draw more reference objects if needed, or background if there's non-specific labeling. As always, you can clear out the reference objects and redraw transfection marker and background For your own samples, press OK. Segmentation options are also available in this step for filtering out small fluorescence or any other background you may have. Clean borders, fills, and split are also. The interactive step is available if you're running a batch process on a multi-well plate or a folder of images. What this will do is, if it's active, we'll stop at that step when you're analyzing images in a batch process. Press the next step button. The data is now calculated, and you can see where the transfected cells are. Here in the data table, we have an estimated count of 160 cells, transfection count of 11 for transfection of 6.8%. If you've modified the protocol for your own sample and would like to save it, simply save all changes to a protocol and press the Save As button. The protocol will show up under the Transfection 1 label with your own naming convention. Next, press the Next Step button, and now we can go through, and if there's additional images to analyze, we can step through them if they're open on the desktop, or if it's in batch process mode, where you have a multi-well plate or a folder of images, we can select those by taking them off the active step, or run on the selected your selected rows and columns in a multi-well plate, or point to a folder and batch process those images within there. When you're done, simply press the Excel button to export